Assalamu alaikum. Today I will present in 5th International Conference on Applied Zoology 2020. The present my topic anti-biofilm and antioxidant potential of green synthesized zinc oxide nanoparticles against the foodborne pathogens. So as you know, foodborne diseases today is a major public health concern. According to the WHO, 30% diseases are due to the foodborne pathogen, especially Escherichia, Salmonella, Shigella, Klebsiella, and Listeria. Since 2000, antibiotics resistance developed due to the foodborne pathogens, especially Escherichia and Salmonella. Antibiotic resistance mostly developed due to the misuse and overuse of the antibiotics, and also due to the biofilm formation and EPS production. So there is a need of the novel and natural antibiotic agents to combat the multi-drug resistant bacteria. Nowadays, we have developed the nanotechnology to overcome the antibiotic resistance by using the nanoparticles. In nanoparticles, metal oxide nanoparticles gain the much more importance in surviving in intensive conditions. Especially in metal oxide nanoparticles, zinc oxide nanoparticles are highly stable and safe due to its application. And zinc oxide nanoparticles used as the biological labeling, delivery of gene, delivery of drug, and biological sensing. And also they are antifungal, antibacterial, antidiabetic, and the acricidal activities. There are many methods to synthesize the nanoparticles, but we use the green synthesis because green synthesis is eco-friendly, cost-effective, and reliable. How we perform the green synthesis of the zinc oxide nanoparticle, in which we've taken the plants, cleaned, iodized, and grind it to obtain the fine powder. The extraction stored at the 4 degrees Celsius for further analysis, and for the green synthesis we use the method of elomeli with the minor modification. Aqueous extraction heated at the 65 to 80 degrees Celsius on magnetic stirrer, and we added the zinc nitrate hexahydrate at 60 degrees Celsius and mixed it stirred for an hour until the white precipitate appeared, and the furnace overnight to obtain the white fine. How we characterize zinc oxide nanoparticles? We characterize zinc oxide nanoparticles by the Fourier transform, infrared spectroscopy, transmission electron microscopy, X-ray diffraction to characterize the shape, size, and the absorption level of the zinc oxide nanoparticles. In vitro susceptibility testing, we perform the three methods: agar-well diffusion assay, evaluation of bactericidal and bacteriostatic potential of zinc oxide nanoparticle, and time killing. Agar-well diffusion assay, in which we perform the conventional method. We swabbed our bacteria on the plate and make the wells of 5 mm and added the zinc oxide nanoparticle in different concentration, and after overnight incubation we noted the zone of inhibition. In MIC, in other words you can say bacteriostatic potential, we noted the we performed the conventional method, in which we make the overnight broth and make the suspension of the bacteria and added the diagnostic nanoparticle and two-fold serial dilution in microtetraplate, and then added the suspension of the bacteria, and then we incubate overnight, and after adding the NBT dye we noted the MIC, and take the 10 microliter from the minimum inhibitor concentration on the plate and spread it and noted the MBC. So in time say were performed in the Mueller-Hinton broth, in which zinc oxide nanoparticle was expanded into the 1 ml of the Mueller-Hinton broth medium containing the bacterial suspension, which turbidity compared to the McFarland solution, and we make the final constant as 0, half, 1, 2, 4, and 8 relative to each isolate, and these culture were incubated for the time interval of 0, 1, 2, 3, and 4 hours at 37 degrees Celsius at 100 rpm, and number of bacterial colonies are quantified as the CFU per ml performed in the triplicate. These all experiments are performed in the in biofilm formation assay. We taken the overnight bacterial broth and discarded these broth from bacterial tubes and attached cell walls with the normal sign. Then crystal violet dye added and excessive dye gently removed with deionized water. Tubes were dried to observe the thin layer of biofilm, and microtitration was used for the quantitative evaluation of biofilm formation, in which we added the 180 microliter Mueller-Hinton broth and expanded the 96 microtitration period and added the 10 microliter of various concentration of 0, 0.25, 0.5 MIC and noted the absorbance after 24, 48 and 120 hours. So next is antioxidant potential zinc oxide nanoparticle. Different concentrations of ascorbic acid and the nanoparticles were prepared in methanol. A color change from purple, violet to yellow was noted and absorb was taken at the 570 nanometer using spectrophotometer. DPH radical scavenging activity was determined by following formula, in which absorbance control minus absorbance sample divided by the absorbance score multiplied by 100. Next, we will come to our results and discussion. Characterization of zinc oxide nanoparticles. As you see, absorption spectrum using the UV visible spectrophotometer, in which 381 nanometer is the maximum absorbance, which showed that there is no relevant absorption pattern is at this absorbance. And XDR beta spectrum show the different patterns which also are relevant to the zinc oxide nanoparticles. Next thing is the FTIR spectrum, in which green synthesizer zinc oxide nanoparticles from the Arabica acacia plants showed. Next is the TEM transmission electron microscope, in which the zinc oxide nanoparticle showing the size and the average size by the XDR spectrum and the zeta potential of the zinc oxide nanoparticle. In vitro susceptibility testing in agar-weld diffusion assay, the zones of inhibition range from the 16 to 30 nanometer. Maximum inhibition was observed against the E. coli at 500 microgram, where the zone of inhibition diameter was 30 nanometer. Minimum inhibitory zone was observed for the Staph aureus 26 at the highest concentration tested. And the MIC value against the Staph aureus, Staph enterica and E. coli were 62.5, 31.25 respectively, whereas the MBC value was for all the 62.5 microgram per ml. These pictures show the time kill kinetic assay. As you know, after the 2 hours for the Staph aureus and the Staph enterica at different MIC is the same time kill, but for the E. coli the time will saturate. And these are effects of the zinc oxide nanoparticles on biofilm, in which the E. coli biofilm eradication more, which is showing in the picture C, as compared to the figure A and figure B, which are showing the anti-biofilm eradication of Staph aureus and Staph enterica respectively. In antioxidant potential zinc oxide nanoparticles, maximum antioxidant potential was observed at the 75 microgram per ml, as you can see in this graph, in which we use the zinc oxide nanoparticle and ascorbic acid as a standard, and maximum antioxidant at the 75 microgram per ml. So let's come to our conclusion. In conclusion, the findings of the present study demonstrate the biofabricated zinc oxide nanoparticles using the Acacia arabica leaf extract effective against the antibacterial and antibiofilm agents against the multi-drug food resistant bacteria. Moreover, the non-toxic nature of the zinc oxide nanoparticles beneficial and biofouling for the prevention of the food contamination. And these all are the references. Thank you so much for your attention.